All right, let's do blood, cardiovascular system, blood one composition. So this is a SEM. What does that stand for? Anybody know? You probably don't. I don't think I told you about this in 201. Well, we talked about microscopes, but I don't think we got into the detail. This is a scanning electron microscope, and it takes 3D pictures of things, and then you can colorize it. So this is actually a blood vessel. So this is the blood vessel wall, this part all around here. This is all blood vessel wall. And then obviously, what kind of blood cells are these guys?
Red.
Red, yeah. And then look at the white blood cells. What do white blood cells do for you in general? Immune system. Yeah, that's good. So when I teach this class at the U of A, we actually get to do this. It's kind of cool. We don't do this. We don't get it from a vein. You just get a finger prick of blood, and then we spin it down in a centrifuge, and we look at how the components of blood separate out. So in this cartoon, it shows you take blood from a vein, put it in a test tube, and then you put it in this thing called a centrifuge. Have you heard of those? Mm-hmm. Okay, they just spin things around. About 5 years ago, my friend from South Korea, mind you, the government contacted her because we had all these little mini centrifuges in the lab, just tiny ones. But because she's Korean and, you know, North Korea uses centrifuges to make bombs, I guess, they contacted her to make sure that she was using the centrifuge for good and not evil. I mean, our government sometimes, because they're just tiny little centrifuges. But we use these all the time in the lab. And in this case, what you're doing is you're spinning it around, and then the heaviest elements, the erythrocytes, or the red blood cells, will settle out to the bottom. And then you'll have the immune cells, your white blood cells, in this thing called the Buffy coat. And then on top, the lightest part, you'd have the plasma. This plasma is mostly water. And then there's other things in the plasma. Does anybody know the other stuff that's in the plasma? Anybody work with plasma and know this?
Electrolytes?
Yes, electrolytes. And give me an example of an electrolyte.
Magnesium, potassium, sodium.
Yeah. Magnesium, potassium, sodium. Perfect. Okay, so then this is really old timey. I guess you get rid of this, but the Buffy coat and the hematocrit, so this red blood cell, the red blood cells are called, that's the hematocrit. What lives in the Buffy coat and the hematocrit? These things called formed elements, and those are the cells. It's like from the 1800s, formed elements, because we know there's cells in there. But anyway, that's what they call it, that's what they say in the book. So when you spin the blood down, you get a ratio of formed elements to plasma. So the formed elements, the ratio of the Buffy coat and the hematocrit to the plasma. Yeah, we talked about that. Oh, I said that you don't have to know many numbers, but when you do, I'll point them out. So you have to know these numbers for the test. So red blood cell percent is called the hematocrit. I just told you that. In females, when you take blood from a female and spin it down, about 37 to 48% of that whole component that's spun down will be the hematocrit, and males is a little higher, 45 to 52%. So that means males in general have more of what type of blood cell? Red blood cell, yeah. Males in general have more red blood cells than females. And then just some cocktail party stats you can talk about. So the total blood volume is between 4 and 6 liters. It's about 8% of your body weight. Plasma is 55%, formed elements are 45%. But then within that 55%, 90% of that is water. Oh, here's some more red. So whole blood has physical characteristics, just like you have physical characteristics. Your blood has physical characteristics. You probably know this. Blood tastes what? How does it taste?
Iron.
How does it taste in your blood?
Iron.
Like ions? Like
Iron.
salt?
Yeah.
Kind of salty?
No.
It has viscosity. What's viscosity? Or you give me an example of viscosity. Thickness, yeah, so like honey, oil, things like that. And the viscosity of your blood is due to the red blood cells. So if you have a whole lot of red blood cells, your blood is viscous. If your blood is viscous, is that going to make it easier or harder for your heart to pump? Harder. So what happens when athletes train at altitude, take out their blood with a lot of red blood cells in it, because at altitude you crank up your red blood cell production. And then they go back down to sea level and they re-inject their own blood that has a bunch of red blood cells in it. What does that do to their overall viscosity? It goes up. And now they're in a race and their heart's trying to pump that viscous blood. It's dangerous. I've tried it before. Just kidding, I know. Yeah, that's. I think that's one aspect of blood doping. There could be other aspects, but that's what I always think of. The cyclists, yeah, blood doping. And they argue it's their own blood, so okay to do it, but it's dangerous. Obviously, your blood has color. It could be more or less red. It has density, pH, volume, and temperature. Oh, look. I see a lot of B's. If you said B, you're right. Give yourself a pat on your back. First cards. Answered right. Okay, so we talked about that. So there are actually 7 formed elements. So there's cells, and then there's these things called platelets. Have you heard of them? Platelets, or I just say platelets, but it's spelled like platelets. What do they do? What do platelets do for you?
Clot.
Blood clotting. Yeah, they're involved in blood clotting. Platelets are not whole cells. Platelets are fragments of a cell. So you've got cells and platelets, or cell fragments. They're suspended in the plasma and they go to the bottom of the tube when spun down. So the formed elements include the platelets, erythrocytes, which are called red blood cells, and then there's 5 kinds of leukocytes. So the leukocytes are the white blood cells. Then we'll talk about those in a sec. White blood cells are immune system cells. We already said that. We're just going to introduce and show you the white blood cells, but when we do the immune system for the next test, we're going to learn what the immune system cells do for you. So what kind of micrograph is this? An electron micrograph, a scanning electron micrograph. Scanning, yeah, because it's 3-dimensional. If it's not 3D, it's not scanning. So here's a white blood cell, and here's a red blood cell. And see this platelet? It's just a fragment. It's a fragment of a megakaryocyte, which is the cell that makes platelets. Question?
What are the platelets?
It's a giant cell called a megakaryocyte, and then that just fragments, that giant cell fragments into cell fragments called platelets. And like if you're a numbers person and you like this, here's the number of red blood cells in a female. What does 10 to the 6th equal? 1,000,000, yeah. So females have 4.2 to 5.4 million cells per microliter. And males have up to 6.2 million cells per microliter. What's a microliter? One what of a liter? That's a milliliter.
100.
Million, there you go. So a microliter is 1 millionth of a liter. Have you heard of this disease, leukemia?
Mm-hmm.
So leukemia is a disease where you just have an increased production of abnormal white blood cells. So it's not just an increase in white blood cells. Those white blood cells are abnormal. They're not fully mature. Oh, 2 B's in a row. If you said B, you're right. If you didn't say B, then get out. I just wanted to quote that movie, Get Out. Did anyone see Get Out? Want to see it? It's crazy. It's awesome. I think it's up for a bunch of Academy Award nominations.
Apparently.
Oh, yeah, same.
Just one?
Yeah,
It's the actor one.
I just read
Oh, the
white
main
blood
actor.
cells and I didn't say low.
That's it.
Really?
I think so.
That's too bad. We should get me on. Okay, so here's table 17.1. This is from your book. It just shows you the major components of whole blood. So mostly whole blood is water, but then it has these electrolytes or solutes dissolved in them. So electrolytes, we just said what those were. They're charged particles. So for example, chloride, phosphate, sulfate, those are all charged particles. And then also in the solutes in your plasma are these things called plasma proteins. And it says here that plasma proteins contribute to osmotic pressure and they maintain water balance in blood. What's osmosis?
Water moving through cells.
Diffusion of water across the semi-permeable membrane, up its concentration gradient or down water's concentration gradient?
Down.
Down its concentration gradient, right. So osmotic, these plasma proteins contribute to osmotic pressure. So are the plasma proteins gonna draw water towards them, or are the plasma proteins gonna push water away from them? Do you remember the rule, water follows salt?
Yeah.
Have you heard that? Well, it's the same thing, water follows solutes. Anything dissolved in water's gonna follow that. So if salt concentration goes up, water's gonna move towards that salty concentration. So plasma proteins are gonna pull water towards them. This is gonna be really important when we look at water balance in the body. I'm just gonna kind of introduce it here. So, for example, if you're, well, it's not if, it is the case that your liver makes the plasma proteins, okay? And the plasma proteins are traveling in your blood, holding on to water in your blood. What happens if your liver becomes cirrhotic and now your liver can't make the plasma proteins? Are you gonna have more or less water in your blood? Less water in the blood. Because the plasma proteins aren't there to pull the water into the blood. Okay, so they're important, and they are, major function of plasma proteins is osmotic pressure. And then also, plasma proteins do pH regulation. And that's even more complicated. We'll talk about that later. But just for now, memorize that. Okay. What else we got here? Oh, this just gives you other things that are in the plasma. Well, we know this. Hormones are always traveling in your plasma because they're traveling throughout your blood right now, trying to find. There's hormones in your body right now. What are they looking for? If they're in the blood and they're not out in the target tissue yet, what are the hormones that are in your blood looking for? Receptors on target cells. Yeah. Yeah. And then you guys know you have oxygen and carbon dioxide in your blood. So. Yeah.
On the last slide, the note, serum and plasma have same, what is serum?
Oh, so this is a table that tells you what's in plasma. All this stuff is in plasma. All this stuff is in the serum, except in the serum there's no fibrinogen.
Okay.
And we'll talk more about this later when we talk about the clotting cascade, because fibrinogen is involved in clotting cascade. I'm not going to ask you a test question on it, but just serum and plasma are the same thing, except serum has one less thing in it, and that one less thing in it is fibrinogen. Yeah.
So are hormones looking to just like physically randomly brush up against a receptor?
Kind of. Yeah. Now you're getting into the world of biophysics, which is really interesting. Yeah. Yeah. And those hormones, if they're in a transport protein, they're kind of popping on and off the transport protein. So yeah, it's a. Pardon me?
To get through the membrane?
Yes. Yeah. Yeah, because if a hormone is traveling in a transport protein and it can never leave the transport protein, then it could never leave the transport protein to go down with the cell. So it's this interesting biophysical kind of popping on and off. Yeah, that's a good question.
So like when you're on the lazy river and you get like out of your.
Yeah, it's like exactly. That's a good analogy. Yeah, growing up in Idaho, I had the most awesome rivers in the world too, inner tube down. So there. Take this. Okay, blood functions. This is a table. You don't have to know all the details, but for the exam, I think there's one question that says, all of these are, it's multiple choice. All of these are blood functions except, and then I'll give one that's not a blood function. So you'd have to recognize that blood functions to transport things. Obviously it's transporting oxygen, hormones, etc. Blood functions to maintain your body temperature. Blood's there to help control the pH of your body, and blood removes toxins from the body, and it regulates body fluid electrolytes like sodium and potassium. And really, when we do respiratory, we're going to talk all about how blood transports carbon dioxide and oxygen. So we'll go into detail on that. Right now it's just an introduction. We won't do a lot about body temperature regulation. We'll do a whole lot about blood controlling pH. When we do the kidneys, we'll do how blood removes toxins from the body. It's super detailed that you'll probably never see again. You won't care about it in the first place. You'll never see again. And then also with the kidney, we'll do regulation of body fluid electrolytes. So I tell you that because I don't want you to feel cheated, like I just give you this list and you're like, oh no, tell me more. Tell me exactly how blood removes toxins from the body. I'm just telling you, relax. We'll go into the detail later on. Blood cells. This is pretty close to what you'll see in the microscope, to tell you the truth. This isn't bad. So let's go over each one of these. So let's start up here. You have to know all these blood cells, you have to know their names, you have to be able to identify them on the lab practical exam. And in a microscope. But if you guys work on the microscopes really hard, then I won't put out the microscopes for the lab practical exam. I'll just use a cartoon. Okay. So let's go over these and then we'll look at them in the microscope. Oh, look, I forgot, they're up here too. Look up here right next to the American flag. We could get those models out. I forgot. Yeah, those are models of blood cells. Probably about $50 a piece. So here's monocyte. Here's monocyte up here. Wait, no, where's my monocyte? Huh, I don't have a monocyte up there. Okay, never mind. So monocyte, what would you say about its nucleus? So it's this purple thing. How would you describe its nucleus?
Crescent shape.
Yeah, you could say crescent shape. the scientists that looked at it said it looked like a horseshoe because they're probably a bunch of old guys wearing horseshoes. Say it's horseshoe shape, crescent shape. Can't think of the other descriptor they use, but so that's the shape of the nucleus. So each one of these cells, you're going to have to think about the shape of their nucleus. And then here, this light blue stuff, this is all cytoplasm. Now, would you say that that cytoplasm looks grainy or granular, or does it look kind of smooth?
Smooth.
It looks smooth. So it would be called an agranulocyte because it doesn't have granules in it. So if you put A in front of a word, it means, yeah, take away. So agranulocytes means the monocyte is not a granulocyte. And then notice its size compared to these other guys. Is it one of the bigger ones, one of the smaller ones? It's one of the bigger ones. Yeah. Okay, next one, eosinophil. So let's first of all look at its nucleus. How would you describe its nucleus? Bilobed. Yeah, that's probably the best way. And then how would you describe the cytoplasm? Is it granular or non-granular?
Granular.
It's granular. And what would you say the color of the granules are?
Orange.
Orange. That's perfect description of an eosinophil. I have a really stupid way of remembering eosinophil because when I was studying them back in the 90s, my nieces were into this cartoon, Winnie the Pooh. Have you heard of that guy? Winnie the Pooh. And there's I think it was a donkey or something named Eeyore, and he had those big ears or something like that. And then that looked, so Eeyore, eosinophil, that's how I remember it. It's kind of stupid, but I mean, we all have ways of remembering things. I'll just share that. I'll humiliate myself and share that with you. Okay. Oh, I forgot to tell you the function. Monocyte travels in the blood. When you have a bacterial infection, it will crawl out of the blood, go into the tissue, and start eating or phagocytizing the bacteria. That's what a monocyte does. Eosinophil is pretty cool because if you have parasites, the eosinophils will all sit on top of the parasite and release toxins and kill the parasite. So it has a slow, writhing death because of the poisons in these granules that will kill the parasite. So that's kind of cool. And then here's the platelets, these little purple things that I said they were cell fragments of a megakaryocyte. And then we have, the next guy is a small lymphocyte. So how would you describe the nucleus in a small lymphocyte? It's huge. It takes up almost the whole cell. And you'll see that in the microscope. If you see a cell and the nucleus is taking up almost the whole space volume of the inside the cell, it's a lymphocyte. And it's the same thing with the large lymphocyte. There you can see a little bit more cytoplasm, but the nucleus is huge. And then if you're a small or a large lymphocyte, what would you say about the cytoplasm? Granular, not granular? Agranular. You can't really see any granules in there. Okay. And a lymphocyte, there's two types, B and T lymphocytes, and they work in the immune system. And we'll go into gory detail about B and T lymphocytes when we do immune. Neutrophil. What would you say about the nucleus? Multi-lobed. Yeah, let's say it's multi-lobed. And then what would you say about its cytoplasm in terms of its granularity? It's granular, yeah. So you can see conspicuous granules in the neutrophil. And neutrophils functions a lot like a monocyte. So it's a phagocyte, so it can go in and eat bacteria. Let's see. Okay. Next one, basophil. Oh my gosh, what would you say about the nucleus of a basophil? Can you even see the nucleus? Not really. But you can really see the big purple granules. If anybody finds a basophil or an eosinophil in their slide, let everybody know because they're hard to find. They're more rare than neutrophils or monocytes. And has everybody heard of an antihistamine?
Mm-hmm.
Well, the basophil is the culprit of secreting histamines in allergic reactions. So histamine comes out of these granules, and then if it's too much, you take an antihistamine. All right. And then the erythrocyte, that's a red blood cell. Does a red blood cell have a nucleus? It doesn't. And what would you say about its shape overall? I mean, it's red. Yeah, it's kind of concave. And it's actually called a biconcave disc. So if you took this erythrocyte and then you turned it on its side, you could see concavity on both sides. Okay. So we're going to identify blood cell types, focus on the nucleus and the granules. We just did that. And then we'll look at the functions, and we talked about the functions. And here's the list of granulocytes, the neutrophils, basophils, and eosinophils. Here's the agranulocytes, lymphocytes, and monocytes. If you want to know, this I learned at the U of A, to memorize which cell is the most and which cell is the least in the blood, has the least number. So neutrophils, if you're looking at a blood slide, mostly you're going to see neutrophils. A little bit less number than neutrophils are the lymphocytes, and then the monocytes, eosinophils, and this. It's really hard to find eosinophils and basophils. They're at like 2 and sometimes less than 1% for basophils. Depends on the blood slide. Yeah, let's go through these and then we can do the microscopes. Guess what this is? Looks like a mountain with donuts on it to me. But it's the head of the pin with, what kind of cells do you think? Just look at the shape. The red blood cells. Yeah, I know it's over here, but you can also see that these would be red blood cells. So they don't have a nucleus, they just have hemoglobin. What does hemoglobin do?
Finds oxygen.
Yeah, it carries oxygen and CO2. Okay, there's no mitochondria, there's no DNA. That means there's no genes, there's no ER, there's no ribosomes, so they don't do any protein synthesis. And they only live for about 120 days. So after about 120 days, the old ones break down. And then, does anybody know the organ that red blood cells, like the red blood cell graveyard? So this organ has really small capillaries, and the red blood cells try to squeeze through those capillaries. They can't get through, they just die in that graveyard. Sometimes you rupture it in a car spleen. Yeah, yeah, good. Is this an electron or scanning electron micrograph? Electron, yeah, because it's not three-dimensional. This shows the biconcave shape of the red blood cell. And then this is a white blood cell, and you can see all these granules. And in this case, they're calling them lysosomes, because they have chemicals in them that will lyse or kill bacterial cells. So here's a snapshot of platelets. So it's just these little cell fragments. What do you think these other cells would be? Can't really tell by their shape or if they're granule because of this picture, but Red blood cells, yeah, because that's mostly what you're gonna see. And we talked about platelets are formed from fragments of megakaryocytes. We'll talk more about platelets when we talk about blood clotting, because they form the platelet plug and they secrete factors for the clotting system so that you can form a nice clot. Red blood cells, we've talked about that, oxygen and CO2 transport on the hemoglobin. They do pH regulation, and we'll talk about that when we do the respiratory system. And we already talked about how red blood cells affect blood viscosity. So covered that.
So we have numbers that are colored. This is stuff.
But it's
that-
not red.
Okay, it's not red.
Yeah.
Okay, so it has to be red.
Yeah, and
Okay.
every time, like there's 3 times the whole semester that you have to know numbers, I'll point it out and tell you for sure it's
Okay.
gonna be on the test. Otherwise, I just put those numbers up there because some people really like numbers. So there you go. I'm always thinking about you guys. So happy. Here's a lymphocyte. This is not a cartoon, this is a photograph of a lymphocyte in a microscope. Check out that nucleus, it's huge. Takes up almost the whole cytoplasm. And you can use, when you're looking in the microscopes today, you can use your slides to see if you have a lymphocyte or not. I mean, you're probably like ballers and you'll just look at it and know it right away, but if you need a little bit extra information, you can use this. So we've talked about the lymphocytes. Here's a monocyte. It gives you the stats on monocyte. It's the largest type of white blood cell. Oh, kidney bean shape. That was the other descriptor they used. So instead of a horseshoe, they said it. Some people said it looked like a kidney bean. And then it gives the color of the nucleus, violet. And same for the lymphocytes, violet. So here's some neutrophils. You can see multi-lobe neutrophils, and you can see that they have their granules. They say they're red to violet. Here's an eosinophil, and I told you that eosinophils phagocytize and destroy parasitic worms. And then I told you basophils secrete histamine. So here's a basophil. If you find them in your microscopes, they'll look like this. So the slides will look like the column on the left-hand side. And then this just gives you the percentages of the white blood cells. All right, if you said C, then you can take a break then. If you didn't say C, no break. All right, yeah, let's take a break. Let's come back at, well, you can come back whenever you want. I'm gonna